So why don't we just, for time efficiency, go ahead with David first, if that's okay with both of you. Again, in the interest of time efficiency, I'm going to go ahead and get going. Welcome everyone to another installment of our Pathways Research Symposia. We have two wonderful speakers here tonight, and we're going to start actually with David Spiegel from Yale. David was a recipient of one of the Visionary Awards. Those are the awards to try to coax people from tangential fields more squarely into diabetes. He got that award in 2017, and a really interesting title of the talk today, Reversing Tissue Crosslinks as a Treatment Strategy for Diabetes. David, welcome.
Thank you so much, Chris, and I'm delighted to see everyone virtually. I remember enjoying, all of our interactions in person way back in the day when people interacted in person, and so this is just a pleasure. You know, I will just start the talk by saying, first and foremost, I'm an organic chemist with a strong interest in the application of chemical tools to solving biological problems, and in particular, developing new treatment paradigms for important diseases. And so that's where I'm coming from here. You're going to hear a lot of things that you probably don't usually hear in these sorts of talks. Let me start by talking about advanced glycation end products. You guys are all probably familiar with these. These are post-translational, non-enzymatic post-translational modifications of proteins that form as a function of glucose concentration and time. So proteins live long enough to see enough glucose to get modified. Those proteins become glycated. These are found in all of us, and not just all of us, but they're found in yeast, they're found in, our evolutionary ancestors. They're probably older in evolutionary time than RNA and DNA. And so it's exciting always to me when I think about the very first time a protein ever saw a sugar, there was glycation. Prior studies in this field before we entered, and again, one of the appealing parts for us was literally, the reagents people would use were these kind of pancake syrup looking mixtures that was literally glucose and albumin sat, on a bench top for 6 months, and super complex mixtures. And yet those mixtures had some interesting biological properties, including evidence for binding to receptors involved in the immune response. And so there are a lot of ways in which ages are believed to impact biology, both in by damaging tissues and also by perhaps in a receptor mediated fashion influencing various kinds of inflammatory pathways. Our goal was to kind of take this mess and clean it up and synthesize specific ages and evaluate how they work. And so when the Pathway Award came along, we focused our efforts on diabetes because, of course, glucose, important predetermining factor in age formation, also, of course, relevant to pathophysiology and diabetes. And so, and I should say, and I'll repeat this later, that there are more than one sort of scientists out there, including myself, that believe that ages, in fact, may be involved in the pathophysiology of diabetes. They're not just simply biomarkers. And so and I can, again, talk more about that. We focused actually on a lot of different ages. I'm just showing you two structures here, glucosapine and the hydromidazolones. We've published a whole lot in this area unrelated to diabetes, and now we've published several papers, of course, thanks to the Vision Pathway Award over the last few years in the area relative to diabetes. But the age that I'm going to talk most about today is glucosapine. I may touch a little bit on the hydromidazolones, but mostly glucosapine. Just going to move quickly here. Again, I told you about simple picture for age formation is you take any protein that lives long enough, expose it to sugar over a long enough time, and you get age-modified proteins. In diabetes, you've got ages that have been characterized, again, that you may be familiar with in eyes, kidneys, cardiovascular system, nervous system, all the places where you see end organ damage in diabetes, and specific links from a sort of diagnostic standpoint. Glucosapine turns out to be among the strongest independent predictors of end organ damage, in particular retinopathy and neuropathy, from out of a sort of, it's an older study, but nevertheless, a lot of reasons to believe that certainly ages are correlated and diagnostically relevant to diabetes, and that they may be pathophysiologically, mechanistically involved. Our approach to studying this problem, again, started from a basic science standpoint, but has moved in this sort of more translational direction, was to synthesize these ages chemically for the first time. They can't really be isolated. They're formed as mixtures and as messes. And when they are isolated, they're isolated in very small quantities. That's how they were first characterized. We want to synthesize the ages. We want to then develop better biological tools to study the ages, better recapitulate the biological systems in which the ages are formed, testing long-held hypotheses in biology and biomedicine, and also develop novel therapeutic approaches. Okay, so let's focus deeply now on glucosapine cross-links. Glucosapine is this weird molecule here that forms on protein surfaces. This happens to be the crystal structure. Anybody tell me what crystal structure this is? I'll give you 25 cents. It's albumin. So it forms on these, it forms on albumin. It actually also forms, probably most importantly from the standpoint of diabetes, on collagen, long-lived plasma proteins. This is glucosapine structure. It's actually, from a chemical standpoint, a very weird, unique, interesting structure. So the chemists actually care about this just at an aesthetic level, as well as its importance in disease. And I got super important about it. Look, I'm an MD, PhD chemist. I'm a sort of rare bird in that sense. And I got excited about this molecule at first because it just represented this beautiful confluence between, the synthetic chemistry challenge and the biological importance. Anyway, it's a cross-link. It forms from a molecule of glucose, lysine side chain, arginine side chain. It's believed to be the most prevalent of the age cross-links. Although we have some reason to believe that that may not be true. I'll talk about that later. And again, it's strongly correlated with retinopathy, nephropathy, and neuropathy. So it's, super important molecule. We went ahead and, and I should just talk a little bit about the chemical interest here. This is what makes this molecule so weird, the 2,5-diaminoimidazole. I'm not going to bore you guys, at least I hope it's not boring. It's not boring to me, but again, I don't want to bore anybody with too much of the details of the chemistry here, but this is a molecule that basically should not exist. It looks like an error. And when I first saw it, I thought, okay, the person who characterized glucosapine made a mistake. This molecule cannot exist. It's just going to isomerize through essentially a hydride shift mechanism to a more stable aromatic compound. Long story short, it does exist. In fact, funny story, when I searched on this molecule, the only example that I could find, the only example I could find of this structure in the entire chemical literature was glucosapine, plus 14 examples of the amino acid histidine drawn incorrectly. So that's how rare this just is, a molecule that shouldn't exist. So how do you make a molecule that shouldn't exist and has never existed before? That was our challenge, or has never been known to exist before. And that was our challenge in synthesizing glucosapine. Again, I won't go through these details. Let it suffice to say that we had to invent a new chemical reaction to get there. That reaction starts from this ketone that I'm pointing my pointer at and ends with this 2,5-diaminoimidazole, getting us from very simple starting materials to a fairly complex molecule that shouldn't exist in a pretty high yield and in pretty short order. And I'll say, that this was very exciting from a chemical standpoint, both the development of a new reaction and the demonstration that this molecule does in fact exist. So we published this in Science in 2015, telling this whole story. It's a great synthesis, at least not to brag, but it's a really efficient synthesis, 7 steps from a known compound, access to milligram quantities of material when previously you could only access, much smaller quantity. We could actually make grams of the stuff if we wanted to. I should also just add that you guys probably are aware the Nobel Prize in Chemistry was awarded today for the development of new chemical reactions that expedite synthesis. Wasn't awarded to us, sadly, for this reaction, but nevertheless, David McMillan and Benjamin List, who won the Nobel Prize, this is, again, not this particular example, but again, why the development of new chemical reactions is so important and deserving of a Nobel Prize is the enablement of synthesizing important molecules like glucosapine. Right, so next we wanted to develop better biological tools, better recapitulate biological systems. I know we're going to run into time trouble, so I'm not going to go through with you how we made this antibody. Let it suffice to say we used our synthetic glucosapine to make an immunogen that we can immunize rabbits with. We did some selections of various kinds. We got a polyclonal serum, and we found, after a lot of hard work, and thanks to support from ADA, antibodies that bind glucosapine selectively. And we did a whole panel of synthetic molecules that look just like glucosapine. It's pretty selective for glucosapine. We published this ACS Chemical Biology 2020. We've done some tissue staining now. For the first time, we can now, anybody can now look, including you if you're interested. We have these antibodies that recognize glucosapine. We can look in tissues. We've given the antibody out now to, I don't know how many collaborators, but we can see here's an example of a mouse aorta. This is time of exposure. These are normoglycemic animals that are exposed over various time periods. And you can see staining goes way up, and it correlates through immunohistochemistry with LCMS experiments, which are very difficult and time consuming, and the way that people previously evaluated glucosapine levels without any knowledge of sort of tissue specificity or sub-tissue specificity. So this nicely supports a link between glucosapine and vascular dysfunction, again, for the first time. Similarly, we think about diabetic retinopathy. We looked at some mouse retina, and we found here big increases in glucosapine in the retinal pigment epithelium, the choroid and Bruch's membrane. It's known that hypertrophy and buildup of protein aggregates in these specific areas are linked to diabetic nephropathy, retinopathy. You guys, somebody on the call may be more of an expert on that aspect than I am, would love to be educated more. But we just started thinking, hey, look, if if glucosapine is causing tissue dysfunction by cross-linking collagen and preventing it from being sort of flexible, and this is and also rendering it immunogenic and rendering it a ligand for rage and so on. This isn't actually our hypothesis. It came from Anne-Marie Schmidt, who you guys may have heard of. But it really makes a lot of sense. If glucosapine and its impact on tissues is contributing to the pathophysiology, boy, what if we could just get rid of glucosapine? So how do you get rid of glucosapine? Well, what we decided to do, and again, thanks to the ADA's help, is develop some novel therapeutics. What's a novel therapeutic? Well, we thought about it. We sort of thought, it was sort of a funny thought process. Glucosapine contains carbon. Bacteria live off of carbon. Anything that contains carbon, a bacteria is going to find a way to eat. I remember reading a great paper by George Church showing that there were certain antibiotic resistant bacteria in soil that subsisted off of metabolic byproducts of antibiotics and antibiotics themselves, which I just thought was hilarious. You have a drug that's supposed to kill the bacteria evolved to figure out a way to eat it. So I thought, this stuff has been around for evolution, since the earliest of evolutionary time. Some bacteria out there must have found a way to eat it. And so what we did was we developed a yeast-based screen of a metagenomic bacterial library. And again, I want to be sensitive to time here. I don't know where I'm at. But long story short, we took a library from soil outside of one of the buildings at Yale, cloned out, and collaborating with Jason Crawford, who does this for a living. This is not my area of expertise. He made this metagenomic library where he was able to clone this library of I don't know exactly how many bacterial genes into a vector that was transformed into E. coli. That, sorry, why is it a yeast-based screen? I think there's an error here, but nevertheless, where was yeast involved here? Anyway, I'm sorry, I'm forgetting this. This was several years ago. It was cloned into E. coli that were oxytrophic for lysine and arginine. And because glucosapine comes from lysine and arginine, we thought, well, hey, an enzyme that eats up glucosapine might release either lysine or arginine, or ideally both. We screened against single oxytropes, but the best results were with the dual lysine arginine oxytropes. This is a super enabling feature about glucosapine, is that we could run this screen. So we wound up finding lots of different enzymes, narrowed down to 4 enzymes. Actually, I have 3 shown here. And lo and behold, we were able to express and purify 3 of these enzymes. 2 of them, SAM and QE, are SAM-dependent enzymes and potentially sensitive to oxygen. We didn't love those. And plus, HEMAF just worked like gangbusters. It really, I have some data if you're interested, I think in the background slides. It works really well to degrade glucosapine. And to top it all off, it has an ortholog that's a human protein. So we found this in a soil bacterium, and lo and behold, there's HEMAF in humans. HEMAF in humans, or an ortholog of HEMAF in humans. HEMAF in humans is actually, it's a separate story that I can talk about in the Q&A if people are interested. An enzyme that's thought to have a totally different function, but maybe has some relation to glucosapine. With our synthesis of glucosapine and these enzymes in hand, we started a company that raised Series A funding in 2019. Seed funding in 2019, and just did another round in 2021, or late 2020. So super excited about the ability to perhaps develop an enzyme therapeutic that could be useful in treating end organ damage. Just as an aside, we used, remember I told you about developing that new chemical reaction to make the molecule that shouldn't exist? Well, like a lot of these ages have that weird molecule, that weird substructure in it. We were able to synthesize this age modic that you guys may have read about in various places, a very well-studied age, in a single step, thanks to that chemical reaction. We published a paper on it in 2019. Again, with your help. And so I'm not going to talk too much about that. I'm going to talk now in the remaining time about pentosinane and pentosidine. If you followed the age field at all, you've heard a lot about pentosidine, especially if you followed it in the early days, back in the 90s, which is when I became interested in it as a medical student. A lot of work looking at pentosidine in diabetes and other and aging and other sort of conditions. Took a long time, but a very good chemist was able to isolate something called pentosinane. His name is Marcus Lederer, and he sadly passed away. I never got to speak to him, but he made a great point, which is pentosidine is probably an artifact of pentosinane, which is the thing that lives in vivo. Pentosidine probably doesn't exist in vivo at all, based on the chemistry. And so I was super psyched just to hear about that. The way that people used to isolate ages was through, boiling a protein in hydrochloric acid, or, exhaustive hydrolysis of various sorts. People have started doing exhaustive enzymatic hydrolysis. Pentosinane may actually even be unstable under those conditions. It's not the most stable molecule under these isolation conditions. And so its significance, its in vivo quantity, pretty much everything about pentosinane, nothing is known. Lederer isolated it and made this hypothesis about pentosidine. We decided to make some pentosinane and test his hypothesis, which none of this is published. So again, I'll be very brief. I won't talk at all about the synthesis. It's a totally different synthesis from glucosapine, even though pentosinane looks a lot like glucosapine. And lo and behold, synthesis is very effective and so on. This is the first opportunity to access chemically homogeneous pentosinane on any scale other than, sort of picogram that Lederer was able to access way back when, after lots of hard work. So we were able to use this now, having recapitulated what we believe to be the real biological system, pentosinane, to test the hypothesis of the pentosinane to pentosidine conversion. Turns out Lederer was absolutely right. Pentosinane converts to pentosidine if you breathe on it. I mean, it's pretty sensitive. I shouldn't say if you breathe on it. Basically, if you do any kind of laboratory manipulation where you change the pH or expose it to oxygen. So we looked at various pHs, various conditions related to oxygen. You'll note pH 6 under O2 free conditions, stuff lasts for days, weeks. pH 7.4 with no O2. Or sorry, this is not no. This is just sort of ambient O2, not rigorously O2 excluded. You get 5.6 days. In an O2 free environment, that goes up to 8.4 days. If you saturate with oxygen, it goes down to 3.1 days. If you look at classical conditions for age isolation, it basically goes away immediately and turns into garbage. So again, highly pH and O2 dependent and destroyed under standard age or historical age isolation conditions. We believe that pentosinane could be much more abundant than previously believed. If it forms in proteins that exist in sort of anaerobic environments at slightly acidic pHs, who knows how long it can last. And in 17 days, if pentosanane is involved in signaling interactions or whatever, it could do a whole lot of biological work, whether that be damage or good or bad, we just don't know. So let me stop there because I don't actually have any idea how much time I've taken, but I'll just summarize and say, I'm sorry, I'm not going to summarize. I'm going to tell you what we're going to do next. We have a polyclonal antibody to glucosapine. We have a lot of it. We want to make a monoclonal to glucosapine. It's actually pretty hard to do. It's taken us a lot of time. We're also in the process of making polyclonal and monoclonal antibodies to pentosanane. Now we have some, and we know it behaves, and I think we can do it. We want to use those antibodies for imaging of pentosanane in tissues. We're developing a new kind of chemical assay to find enzymes that break pentosanane because it's probably too unstable for our sort of metagenomic assay. We've actually spent a lot of time making synthetic collagen that contains glucosapine. We're not there yet. It's really hard to do chemically, but once we have that, we'll be able to actually measure for the first time what the biophysical impact is on collagen of this modification. And finally, on a totally unrelated note, because of the wonderful, thank God, unrestricted sort of nature of this award, we just started getting into diabetes and making friends with the people involved in diabetes at Yale. And Kevin Harold and I have become friends, and we've come up with what we think, we've used some of these funds to come up with what we think is a killer way to use CAR T cells to eradicate type 1 diabetes. I don't know how much of it I'm allowed to share. I didn't ask. I don't know if we filed on it or anything like that. I just wanted to mention it because I'm super duper excited about it. And I think it represents, at least in part, what this award was designed to do. So these are the people that did all the work. They're wonderful. I'm grateful to them and to you. Thank you for your time. I'm happy to answer any questions. I hope, I don't know, am I early or late or where?
No, we got a few minutes for questions. It was a fascinating and very clear talk. Let me start with an outsider's question. Does glucosapine cause lysine to arginine cross-links across proteins or only within proteins? And then a second question that I have is, when you use your glucosapine busting strategy, are you at risk of damaging the lysine or arginine residues on the protein when you bust the cross-link?
These are wonderful questions. So question one, keep in mind, it's the glucose that becomes glucosapine on the lysine and arginine,
yes,
not the glucosapine that causes the, so maybe I didn't understand your first question.
Yeah, no, I'm just, so if the lysine and arginine are close to each other and you're in a hyperglycemic state,
yes,
then glucose serves as the covalent connector, if you will.
Correct.
But I don't see any reason why that can't occur to two adjacent proteins.
Oh, of course it can.
Yeah.
In fact, I'm sorry, I totally missed
Yeah.
it. Yes, that's what happens in collagen. Look, collagen is triple helix, right?
Yeah, okay. All right.
You got interstrand crosslinks
Yeah.
that are the big problem.
Okay, I got you.
I
Okay.
think it's happening all over. So yes, 100%.
Okay, and your busting strategy, is there any risk of damage?
Busting strategy, absolutely. There's risk of damaging lysine or arginine.
Okay.
Heme F seems to leave it gives you back, it's a little bit, so it gives you back ornithine and not arginine.
Oh boy.
And it's a little, we all know that ornithine is a problem in celiac sprue, I believe, right? And there are drugs out there that, are,
right.
It's an autoantigen essentially. We're not there yet with HEMF. There may be conditions under which HEMF gives you back arginine. I'm not completely sure. You know, it's tantalizing to believe that HEMF is in humans as an enzyme designed to break glucosapine, but I just don't really think it is. The question is there, you know, is there a market for, anyway, there are some therapeutic issues still to be resolved. I don't think that these enzymes are perfect. And you're right, you know, there's sort of an, you know, there's an adage in drug screening. It's you get what you screen for, right? We didn't screen for, and well, I suppose we sort of did screen. We wanted to get back both intact lysine and arginine, but it turns out that these auxotrophs were able to survive on the ornithine that was produced. I think they had a way of converting the ornithine into arginine, right? Which makes sense. That's what's happening biochemically.
Let me make, there's two other questions we're going to get to. So Andy Stewart, beautiful chemistry. How might you envision using HEMF analogs as drugs? How do you envision using HEMF analogs as a drug, since so many proteins and so many organs are glycated? And might it be useful to think of a small molecule that blocks glucosapine pentosane formation?
All great questions. I think the answer to one, using HEMF analogs would require making an analog that sort of changes its mechanism in such a way as to keep the arginine intact. Not clear that HEMF is going to be a drug. How do we envision using them as drugs in so many proteins? So, you know, that's a very good question. I think it's a drug delivery question. I don't know that we're going to have an IV administration of HEMF, although frankly, there's really no evidence to suggest that glycation is ever good. So if organs all over the body are glycated and HEMF goes all over the body and gets rid of glycation everywhere, that actually just may be good. Glycosylation, of course, is important, but keep in mind the difference. Glucosapine, now I have a pet hypothesis that glucosapine is doing something important biologically, and that's why evolution has allowed it to stick around, but there's no evidence for that. And
okay,
so,
I think we're going to have to move on.
Okay, I'm sorry.
No, you shouldn't be sorry. These are it's fantastic stuff, and we're obviously all very interested. Marlon Pragnell asks, do you see increased levels of glucosapine bound to people with diabetes? And Alan Addis asked, does the addict with albumin interfere with its ability to bind to fatty acids? And if you don't mind, David, if you would answer those in the chat, so we and go ahead and we can go ahead and switch over to Steve Parker's slides.
Do you mind if I just make a call to the group and say, hey, if you guys have access to samples from diabetic patients that you're willing to give to us for us to study, contact me.
Absolutely. But please, if you don't mind, address the two remaining questions in the chat, and let's get Steve's slides up here. Thank you so much. That was really fascinating.
Thank you for everything.
All right, Steve.
Hey, can you see my slides now?
We can see it and hear you. Wonderful.
Okay, wonderful. Let me turn the laser.
So this is Steve Parker from Michigan. His award was an Initiator Award in 2014. He's completed his Pathways Award, but I know there's a lot of very productive work that's gone on. So Steve, take it away.
Yeah, thanks, Chris, for the introduction, and thanks everyone for attending. It's really exciting to be able to share our work with everyone here. What I decided to do is give you a preview of some ongoing studies. So these are kind of hot off of the instruments and hot off the analyses, and none of this is published yet. So I hope you enjoy it, and I would love to hear some feedback about it. And the theme is convergence on chromatin and diabetes and how we use single cell multi-omic profiles to nominate gene mechanisms at GWAS signals. I just wanted to briefly introduce the lab. This is kind of how we exist in this hybrid nature of scientific research nowadays, where the computational side of the lab over here on the left and the wet bench side of the lab over here on the right, with a number of staff level PhD folks and some postdocs and some PhD trainees. We actually have another postdoc joining in January from Steve Rich's lab, and we're looking for more postdocs. So if any of this looks interesting to anyone that you know of, please send them our way. We would love to talk. Most of the work I'm going to present today is a short story led by Ricardo Albanes, who was a PhD student, then a short postdoc. Now he's doing a postdoc at WashU in St. Louis on T1D, and Arushi Varshni, who's leading more of T2D work, data generated by Nandini Manikam. So I'm going to start off to try to motivate the biological complexity across dimensions and emphasize why I think chromatin is such a important molecular layer. Then I'm going to tell you two data stories about how we're using single cell multiomics to dissect GWAS signals. The first is dissecting type 1 diabetes GWAS signals and pancreatic islets. The second is T2D and related trait, and I'll show you some really interesting, I think, waist to hip ratio data in skeletal muscle. The T1D project in islets is across a dozen samples, and the T2D skeletal muscle project is across hundreds of samples, along with some conclusions, and hopefully there's some questions that I can actually answer. Oops, wrong direction. So diabetes, genetic predisposition starts down here, this kind of foundational layer, your genome. And these genetic variations, there are hundreds throughout your genome for both T1D and T2D and related traits, percolate upwards through these molecular layers, influence the epigenome, transcriptome, proteome, et cetera, all the omes you can think of, until finally you get this complex phenotype that we call diabetes. The really interesting thing, I think, to me, is that your genome is essentially invariant in terms of risk for diabetes and common alleles. We're not talking about somatic variations for cancer here. We're talking about germline inherited variation. All of the other layers are very context specific. So they change depending on what cell type you're in, and they change depending on which cell state that particular cell type is in at any given moment. So I'd like to put a gold star next to the epigenome, because I think it's the most proximal layer to most of these variations that predispose to disease. All of them are, not all, but 90% of these SNPs are in non-coding no man's land. So likely they're influencing chromatin architecture, which then gets propagated to a gene expression and on upwards through this dense molecular network. So we really focus on trying to interrogate that molecular layer. The first project, looking at T1D and pancreatic islets, we've collected 6 healthy controls, 3 autoantibody positive individuals, and then put them through joint single cell RNA-seq and single nucleus ATAC-seq profiling on the 10x platform, and then done lots of computational analyses. We've also taken 3 of these healthy individual islet donor samples, and then either done a control exposure, a cytokine cocktail exposure, or a CVB4 infection exposure, and then done subsequent single cell RNA and ATAC profiling with the associated computational analyses. Just due to time constraints, I think I'm not going to talk about this today. I'll focus mostly on these folks. Here's what the data looks like. This is a little over 100,000 post-quality controlled cells or nuclei, about 50,000 ATAC, about 70,000 RNA, and you can see all of the clusters that you would expect. For the islets, we see a single alpha cell, a single beta cell, delta, gamma, and we see ductals and acinar and other cell types like you would expect. And here are the different modalities, ATAC on top and RNA on bottom, which basically shows that we can integrate across these modalities without having any modality specific batch effects. Here's a closer look at the data, just showing some of the markers to give you a little bit of confidence that we're assigning the right cell type. So here are what we're calling beta cells, and you can see they're very high for insulin expression, alpha cells high for glucagon expression, et cetera, across the diagonal. And here's the single nucleus ATAC data showing the corresponding open chromatin accessibility profiles at the locus. So here's the insulin locus, and you can see massive open chromatin accessibility in the beta cells, lower in everything else, et cetera, across the diagonal. Now, once we want to look closer at type 1 diabetes GWAS variants, here again is the insulin locus, and I'm showing you beta cells at the top and all the other cells below, and I'm showing you ATAC-seq tracks here. You can see the top track here. These are SNPs associated with type 1 diabetes, and this is one signal at the insulin locus, another signal at the insulin locus, and yet a third signal at the insulin locus. So 3 independent signals of association here. And what Ricardo Albanis, the postdoc that was leading this project in the lab, did is developed an algorithm to look across these SNPs and simply just quantify the extent of chromatin accessibility, and then weight that level of chromatin accessibility by the probability of association. And then you can kind of display what's the most likely causal cell type underlying this particular genetic predisposition. And here, unsurprisingly, at the insulin locus, the beta cells really light up and jump off the page, unlike the other cell types. What we can do is look across all 136 reported in this recent Chu et al. Nature 2021 paper. Here across the x-axis are the 136 different signals that predispose to type 1 diabetes. And then here's that quantification of the chromatin accessibility across each of the cell types, weighted again by the genetic predisposition of the individual SNPs. And as one might expect, there are a lot of this cluster of immune variants with all the IL associated signals, like not surprising. And then over here, what was I think more interesting to see is there is a beta cell component, and in there, here's the 3 insulin signals, and then a number of other signals that look like they're really specific to beta cells, or maybe in general the islet components, the endocrine component of the islets. So what we want to do is try to look closer and see if we can nominate target genes for these, non-coding, usually gene distal signals. And I'll focus on these associations here: TOX, RAS-GRP1, and DLK1, which have 2 different signals. Looking at the TOX locus, again, here's the beta cells at the top, alpha, delta, gamma, et cetera. You can see chromatin accessibility in this GWAS region is higher in the endocrine islet cell types than the acinar ductal and other cell types. And here is what looks like one of the most important SNPs. And what we do is this analysis to infer 3-dimensional looping using chromatin co-accessibility across the single nucleus ATAC profiles. And this peak right here that overlaps the SNP looks like it loops over and interacts with the promoter for TOX. So we've established what might be the causal regulatory element, what might be a causal cell type, and what might be the causal target gene for this T1D GWAS signal. And one of the things that we do to build on top of this is do functional fine mapping. And you can see here the probability of this SNP just using genetic data is pretty high, but then it gets even higher when you use functional fine mapping approaches to include the single cell ATAC-seq data to try to figure out what's the causal SNP. We then make these computational models. We use a support vector machine to identify important sequences in the ATAC-seq data, and then use those to predict the effect of the risk allele at that particular SNP. And you can see here, the risk allele looks like it increases chromatin accessibility. So what we're doing right now is in collaboration, and I'll acknowledge this on the next slide, with Xubing Chen at Weill Cornell. She's already created knockouts and iPSC lines at the TOX locus that I described, and the other 3 loci that I didn't show here, for both the gene and for the regulatory elements that are harboring the non-coding risk variants. Then those are being differentiated into beta-like cells, and a number of assays are being done on that material. So I want to acknowledge Xubing Chen at Weill Cornell. This was led by Zuming Tang in her lab. Francis Collins was a postdoc in his lab. We still collaborate with him. Henry Taylor helped do some of the RNA analysis for this project. And Ricardo Albanis, the postdoc in my lab, is the one who's been leading this. So I quickly want to switch gears and talk about diabetes from a different perspective. So this is insulin resistance in skeletal muscle and related traits of diabetes. So it's a similar project where we're doing multi-omic profiling, so RNA attack in skeletal muscle. And these are the types of cells that you expect to see in a skeletal muscle tissue biopsy. This is what I'm going to present today is one part of a larger project. So here we did this, we got 287 skeletal muscle biopsies across individuals from the fusion cohort. In the larger study, we have 408 skeletal muscle biopsies. What we did is isolate nuclei from those. We mixed these samples in batches of 40, and then took part of the nuclei and did single nucleus attack-seq. The other part did single nucleus RNA-seq. And we can assign which individual the nucleus belongs to based on the pattern of genetic variations in that individual. Then we do a bunch of data integration and clustering to figure out what are the fiber types and endothelial cells and satellite cells and all of that. And then finally, what we can do, what I'm really excited about, is integrate genetic variation. So here are the 3 possible genotype classes on the x-axis with both of the molecular traits, chromatin accessibility and gene expression, to identify eQTL or gene expression quantitative trait loci that are associated with genetic variation, or CAQTL, chromatin accessibility QTL, and then compare those to GWAS signals to try to infer what are the mechanisms at a GWAS locus, what are the cell types, what are the regulatory elements, and what are the target genes. So this again is an example of going from genome to epigenome to transcriptome in a chain of causality, hopefully to see what influences this complex trait. So here, I'm going to skip through this really quickly. We did this experiment in 10 batches. You can see across the columns. And the modalities you can see in blue is the attack on top, and in orange is the RNA on bottom. And these UMAPs essentially look the same across everything, indicating that we don't have modality-specific batch effects, and we also don't have batch effects across the 10 experimental batches that we conducted. Collectively, these 287 samples represent over 400,000 PAS-QC nuclei. And these are the cell types that we identify, not surprising. And this is the abundance that they occur in. So about 60 to 70% of the skeletal muscle biopsies are made of muscle fibers. These are, most abundant, but they're also multi-nucleated. So we can see type 1 fibers, type 2A and type 2X. And then we see this long tail of lower abundant cell types. So mesenchymal stem cells and endothelial cells at mid single digit percentage representation, all the way down. We see neuromuscular junction. It's really cool. All the way down to adipocytes at less than 1% of all the cells. You can see a very clear adipocyte cluster right there. So the next thing we want to do is take the most abundant cell types and do those eQTL, expression genotype association scans, and CAQTL, chromatin accessibility genotype association scans that I was talking about, and compare those to GWAS signals. So we identify across these most abundant cell types thousands of eQTL and CAQTL. So here's the number of eQTL we identified at 5% FDR, and the number of CAQTL that we identify, an order of magnitude or more larger, CAQTL than eQTL. And again, we're looking at CAQTL connect genome to epigenome, and eQTL is a way to connect genome to transcriptome. Then we can perform co-localization across eQTL and CAQTL. So that's comparing across the layers. If an eQTL and CAQTL co-localizes, it means the same genetic variant is involved in genetic regulation of chromatin and genetic regulation of the transcriptome. And so here's a bunch of genes that have eQTL, about 1,400, and the cell types that they are represented in here on the y-axis. And what I'm showing you is if a CAQTL co-localizes with the eQTL for that gene, and this is just plotting the strength of that association. Anything above 0.5, which is posterior probability of hypothesis for colloq for any aficionados, is really strong indication that the same genetic variant is controlling chromatin accessibility and gene expression. And the really cool thing about this is you can see most of the co-localizations are cell type specific. So there's this genetic regulatory architecture that's unique to each of the individual cell types and not shared across the cell types like over here in this part of the plot. So we wanted to look closer at one of these, and I'll show you an endothelial cell example. And remember, this is present in maybe about 5-6% of the nuclei that we pass QC. So here's the attack-seq track. In aggregate, here's what you would see if you didn't do single nucleus profiling. So nothing there, totally boring. Fiber types are totally flat. Here's endothelial cells. You see a nice ataxic peak in the endothelial cells. Here's what it looks like if you partition by genotype. So homozygous A individuals in dark blue, heterozygous individuals in light blue, and homozygous G individuals in gray. It's clearly a very strong CAQTL there, where genetic variation influences chromatin accessibility. The really cool thing is now this CAQTL, this is a locus zoom plot where I'm plotting on the y-axis the negative log 10 p-value. Each point is a genetic variation. Here's the lead CAQTL SNP right here. This SNP is also the lead SNP for an EQTL for TMEM150C. So this peak is nowhere near TMEM150C, but genetic regulation of chromatin accessibility is also associated with expression regulation of this particular target gene. So it's a way for us to go from a genetic variant in the genome to the chromatin that regulates, and then finally to the target gene that it regulates. So now the even more powerful approach that we've recently started using is to invert the direction of causality between this. So in other words, is the chromatin influencing the RNA, or is the RNA influencing the chromatin? And you can use this Mendelian randomization approach to dissect those two different possibilities. And so here's the first possibility, one of three possibilities. So here in vertical is a cartoon of a SNP. In orange is a chromatin accessibility peak, and in green is a gene. So the first possibility I'm describing is the chromatin accessibility peak causally influences gene expression. And it's illustrated by this SNP influences the peak, and then the peak influences the gene. So that's chromatin causal on gene expression. You could also have gene expression causal on chromatin accessibility, right? So SNP is associated with gene, but then gene is associated with peak accessibility. And then the third possibility is just, we don't know what's going on, horizontal pleiotropy. The SNP is associated with each of these different molecular traits, and there doesn't appear to be a causal direction between the two. When we quantify each of these, we get thousands of causal associations and a few hundred that we're unable to determine. So let's go back now and look at GWAS variations. So for the skeletal muscle, we've looked at lots of different traits. Type 2 diabetes is interesting. Waist to hip ratio is another GWAS, T2D related trait that I think is very interesting, and we detect lots of co-localization. So here's the number of EQTL co-localizations we detect in the different cell types, and here's the number of GWAS CAQTL co-localizations. So lots more CAQTL than EQTL co-localizations. And then we can look at how cell-specific these are. Again, there's a large chunk of these co-localizations that look like they're operating in distinct cell types. Here's one example in mesenchymal stem cells. You can see nice ATAC-seq data there. You would never see this if you did bulk ATAC-seq in skeletal muscle. It's a really nice effect of genetic variation on chromatin accessibility. And then here is the CAQTL plot, nice lead slip there. And here's the GWAS plot for waist to hip ratio. This peak occurs in an intron of the EBF2 gene, which is implicated in controlling pre-adipocyte cell identity. And I'll note that this is a precursor cell type, suggesting perhaps this represents a genetic predisposition at an early stage of development where the MSC might differentiate in one trajectory or another. We don't have evidence for that, but it's something we'd love to follow up on. Here's another locus where there's nice co-localization. Here, this is clearly in muscle fiber types and not in any of the other cell types. It's a beautiful CAQTL. And here is the CAQTL. It's at 10 to the minus 40 p-value. Here's the EQTL that co-localizes to, for this gene, SEMA3C. And then here is the GWAS signal for waist to hip ratio. Now we can do our causality inference approach. And for this one, this Steiger p-value is 2.3 times 10 to the minus 5 for causality of the SNP affecting chromatin accessibility, and then chromatin accessibility affecting the gene. So it takes a causal trajectory of the proximal event is chromatin accessibility is affected by genetic variation, then gene expression is affected, and then subsequently the trait is. SEMA3C is an adipokine, interestingly, involved in exercise-induced improvements of metabolism. So I'll quickly summarize by saying, I hope I've emphasized that in terms of genetic predisposition, diabetes and related traits are diseases of chromatin biology. Most of this is genetic variations, fine-tuning regions of the non-coding genome. Islet single-cell multiomics nominate beta cell target genes for T1D. Skeletal muscle single nuclear RNA tech profiling provides this rich data set that allows us to identify targets in even rare cell types. And one of the things I want to emphasize is that there's many more co-localizations of trait GWAS with CAQTL than EQTL. Again, I think providing more evidence that chromatin is a critical layer to understanding diabetes and related complex traits. And many of these CAQTL co-localizations are cell type specific, indicating that you really need to look at the single cell level and not the bulk level if you want to understand more precisely which cell types are involved. I'll end by thanking a really diverse team, folks in my lab. We collaborate closely with Mike Binky and Laura Scott here, still with Francis Collins and ex-Collins colleagues like Michael Stitzel and Praveen Sathapathy, who are also Pathway awardees. There's lots of other fusion collaborators, additional collaborators to help get some of those 408 skeletal muscle biopsies that they sent to our lab. We are very fortunate to be supported by NIDDK, the FNIH, ADA, and we're looking for postdocs. And just as a heads up, for anyone attending ASHG, this is the American Society of Human Genetics, happening in a couple of weeks. We're fortunate to be presenting 5 different projects there, 3 platform presentations, one by Ricardo, one by Arushi, and one by Adelaide, and then 2 posters, one by Peter and one by Krista. So if anyone's attending that, these folks can explain the projects way better than I can. So thanks to them, and thank you all for listening.
Well, thank you, Steve. That was pretty dazzling. That was really well done. Thank you.
Thanks.
I would love for someone who's more chromatin literate than me to ask an intelligent question. Alan Addy or Praveen or Paul Cohen, any of you guys want to step up and ask something of Steve?
I have a question, Steve. That was really, really nicely done. Are the regions that of these SNPs, are these SNPs that are cross-linking or that you can bridge to these promoter spots? And this goes back to the first part of your talk. Are those particularly well conserved? And do you see, for example, conservation with of those loops with the looping structures that have been determined in mouse islets?
So that's a great question. I don't think we've looked at that a ton recently. I think what's more important than the evolutionary conservation is just the analogous conservation, or the conservation of the analogous of chromatin architecture and the analogous cell type. So I think Michael Stitzel had a really nice paper that we're a co-author on in the American Journal of Human Genetics a few years ago, where he looked specifically at the C2CD4A and B locus, and we found conservation of chromatin architecture there. We've done, in collaboration with Chris Newgard, this is a few years old now, and we still need to publish it, but it's quite striking and really cool results, but just not written up yet, where we looked at rat islets, and we've done chromatin profiling there, and we found that if you take those enhancer regions that you identify in rat islets and project them into the human coordinate system, they are highly enriched to overlap T2D and related trait GWAS elements. So I don't know about evolutionary sequence conservation. To some extent, probably so, but certainly in terms of functional conservation of chromatin and analogous cell types across species, there's definitely some conservation there. And apologies, Chris, we're going to get that published one day.
So are you going to go ahead and delete some of those in either rat or mouse?
Yeah, so I maybe not deletion, but we're part of a team that's doing a CRISPR I screen to survey those kind of elements. Yeah.
Any other questions? Michael and Praveen, you're collaborators, but that doesn't matter. Any questions that remain?
Chris, I was actually going to highlight a question that Stitzel asked in the chat because I was curious about the same. What do you think, Steve?
Oh, hold on, I didn't see that.
Yeah, so I can just paraphrase. So it looked like most of the T1D SNP overlaps were with like resting islet profiles, right? So I'm just curious, was that the theme? We didn't get to see like the CVB and the cytokines, but do you find ones where it's like a real context specific and where you say it's like the islet function when they're responding to cytokines or CVB infection?
Yeah, that's a great question. So I didn't show that data, sorry, and I did that in the interest of time. I want to talk about a few projects, but we do. So what we do is, look at the attack data under basal versus under these different perturbation conditions, and then go back and look at the enrichment. and for GWAS signals. And the thing that was cool, and I should have shown this, but the enrichment actually increases for GWAS SNPs if you look at those perturbation conditions, particularly for beta cells. Under basal conditions, unsurprisingly, the immune cells are the most enriched for T1D. But under those perturbation conditions, the beta cell enrichments look like they start increasing, maybe not quite as high as some of the immune cells, but definitely higher than basal conditions, suggesting that, yeah, it's really, I think it's going to be context-specific regulatory element activity that is driving these associations. And that's another, I think, big reason that chromatin is so important. I think chromatin is kind of like the history book of what the cell's been doing, but it's also represents the potential cell states that cell can occupy.
Sorry, Mike, glucosapane busted up our chat. So.
No problem at all.
Anyway, this was absolutely inspiring. Thank you, Steve, and thank you, David. Great talks today. November 3rd, same time, Wednesday, 2 p.m. Sui Wong and Alexander Kostic. Please join us next month, November 3rd. Peace and love, everyone. Good job. Thanks.
Thank you, guys.
Take care.
Great
Thank you.
job, Steve.